In this room, we're going to have a set of lightning talks specifically related to some newer projects here at Macerk, starting with Dr. Ben Minerich, who's going to be presenting on culturing microalgae to support aquatic invasive species control research. So, Dr. Minerich, take it away whenever you're ready.
Hi. All right. Thank you. Thanks everybody for coming to join on this. Yeah, so I'm just going to be kind of sharing an update of this project, which is tied to a few other projects throughout Mazurk. So give you a little insight to what I've been doing over the last year. So as everybody knows, invasive mussels have significant implications, not only just to ecosystems and to the animals around, but a big financial burden in terms of intake pipes being cleared from intake pipes, both for heating and cooling, but drinking water as well, and effluent and connection between different water bodies throughout the state. So to the tune of about 60 million annually is different control methods right now for just removing these animals mechanically or combined with different chemicals. So my project is working to on a kind of couple different angles to support some different AIS control. One of which is you'll hear about in the next session, I believe, from Dr. Gold, that he's working with some RNAi interference. And in that aspect, we're working with just kind of supporting keeping adult zebra mussels alive in the lab for a lot of experimentation. And then another project which will have future implications, taking care of and rearing young larvae of different zebra mussels. So right now that is another project that I'm involved in. We're targeting these small larvae, so getting adult broodstock from different areas around Minnesota, inducing egg and sperm production, fertilizing those, and then trying to grow those up, which will give some opportunities for biogenetic control methods. And at this stage right here, the trochophore larva is when they start to eat. And one of the major uh, kind of bottlenecks in this rearing is food and nutrition. And so talk a little bit later about how that influenced some of the choices we made going forward in this project. But I had been using some other combined or some concentrated marine algae historically to feed just adults. And as you'll see in this next slide, actually found incorporating live algae had a greater impact on survivorship and growth. Here, Dr. McCartney had done some research back in 2021 using shellfish diet, which is a marine algae, and just feeding that. This is commonly used in a lot of bivalve aquaculture. But then also at the top, these red dots, just feeding live Isochrysis galbana is another marine algae that he was culturing in the lab, found with the larger individuals had significantly higher growth. So we had been working also with a consortium of different individuals working on quagga mussels, zebra mussels, and the golden mussel, all of which share a pretty similar life cycle. And they had also had similar experience with live algae being crucial to kind of making sure you could grow these zebra mussels in a lab to make sure that they were nice and fit for good results in terms of kind of as you're as you're trying to measure whether or not you're effective at killing them. You want to make sure that they're not just naturally dying. So where we started first, Dr. McCartney had kind of thrown up some research on some of the fatty acid profiles and trying to find correlations with overall kind of fitness or growth and survivorship. And this being a saltwater algae, there is another confound that you're always introducing new salts into the water, and we wanted to make sure that we were isolating more of a freshwater algae. So we looked at a number of different candidate species of freshwater algae. We settled on the Chlamydomonas reinhardtii, Chlorella vulgaris, and Nanochloropsis limnetica. As you see, they kind of fill out similar fatty acid profiles to the Isochrysis galbana. And so we wanted to be able to offer that up to these animals to make sure that they had a nice, well-bound or well-rounded nutritional profile to be able to rear these animals. Here are those different algae species under a microscope. And another component that made Nanochloropsis limnetica in particular valuable is that it's much smaller than the other two species. So in the project I'm working on to try to rear these animals in captivity, this is a good candidate species for a first food for those young larvae to be able to feed. So majority of this last year was spent building a lot of infrastructure. We have growth chambers at Mazurk. We can control lighting and humidity and day length and light output strength. And so worked on finding a good candidate environment to grow a lot of these algae. And here you can see some Chlamydomonas and some Nanochloropsis growing in the background, that lighter yellow, on these shaker plates, which keeps the animals, the algae suspended so they can grow nice and dense. And then building out a grow room in one of the BSL labs. So this is a grow light, which we can match peak output light frequencies to make sure that we're maximizing energy input to what we're going to be outputting for light that's usable for those algae to be able to grow. And there's 3 of those carboys right down there right now. There's probably 5 times as many growing in the lab currently. So one of the other first challenges that we had was trying to figure out how quickly we could grow these algae. So the Chlamydomonas grown in 1 liter, we use special media, which is outfitted with a lot of nutrients, high in phosphorus and nitrate and acetate in the terms of the Chlamydomonas and the chlorella that they use to grow, as well as the light. And looking at these growth curves, actually found that in order for us to get a big volume of algae cells grown, could take almost up to a month once we transfer these up into a larger 190 liters. And so work to find right in the middle of this growth curve here at day 5, when the algae are growing through an exponential growth. That's the best time to move them up to larger carboys and then up to those larger 190 liter tanks. We found that we were able to get everybody on about a 5-day schedule to maximize algae growth and providing those essential nutrients. And so we moved on to about a 3-week cycle in which we're able to grow and then harvest algae. And so we kind of figured that out over the last year, and also worked on a lot of methods to maximize sterilization to prevent any sort of opportunistic animals and ciliates from growing in those algae cultures. So we've got a process down now. We've been working over the summer to try to grow these algae. Use an oil centrifuge to harvest from those large growth tubes and centrifuge down to concentrate, which is really beneficial. Then we can do cell counts under the microscope, and then we can meter this out based off of volume to get a controlled amount of food that we're feeding out. So right now we're in about the 2nd week of our project. The zebra mussels on the left are all individually tagged, measured by length, and then weighed. We do a dry weight on those. Well, kind of basically a wet weight, but we let them open up and drop their water, so we're not also including that water weight. And then we have them divided in on the panel on the right. You can see there's 4 different treatments. The first 3 are isolated by just 1 algae species, so we can look at limitations of specific amino acids that might be present in those algae. And then the 4th one is a combination of all 3 algae species, so we can see what kind of concurrent benefits may be coming from those algae. So we'll be measuring these animals over the next 2 months to see what the best algae is for growing these animals as adults in the lab for other future studies, and then be using some of that algae also as we're doing continued analysis on trying to grow some of the larvae. So in wrap up, just wanted to thank all the algae techs, Sarah, Anna, Emily, and Abby, and then Mike for being a partner on this, the Goal Lab, and Jay for managing things out at Mazurk, and the Minnesota Zoo and Mazurk for helping to support this work. So yeah, with that, we'll wrap up. I think, do we go to questions right now, Alex?
Yes, we do. Okay. So I will pop in here to remind folks that we are using the Q&A feature here to coordinate Q&As. So if you have a question for Ben or any of other speakers, that's where we'd like you to be dropping it. And it looks like we've got some things that have already started to come in here. I'll start with this, Ben, to just give you sort of a warm up here, considering I thought this was a good question. You know, thinking broadly, generally, you know, maybe less scientifically mindedly, what would you sort of say to an audience of people who find it surprising or ironic or confusing that a species that is so easy, so abundant out in the field where we literally can't stop it from growing, is so difficult to raise in a laboratory setting or in a research setting. How would you sort of reconcile that reality, given that it's maybe not intuitive for most people?
Yeah, just with humor. We have significant limitations on this project that are all extremely humorous. With how invasive and how present zebra mussels are in the Twin Cities area, there's no really good place for collection where consistently we can find large individuals based off of that they need to grow on top of, riprap or rocks, hard rocky bottoms. And so we've tried. I've been working on this project really for about 3 years, and the first year was going all around the Twin Cities, Lake Minnetonka, up to White Bear Lake, trying to go up in kind of some North Lake Chains, like Fallon area, and not a lot of good opportunity just for harvesting algae or harvesting zebra mussels. This year in particular, 2 years ago we had really good success with spawning, inducing spawning. This year we haven't had a lot of good females producing eggs, so it's one of those bust years. That's the year that we finally had all the infrastructure ready to kind of grow these animals. And then once in the lab, that's working with this worldwide group, down individuals from Brazil, Michigan, Missouri, that are all trying to raise these different animals. And it's a very sensitive early life stage that even though things like blue mussels or oysters, a lot of that aquaculture technology has been developed, doesn't seem to translate directly over to native zebra mussels or invasive zebra mussels. So a lot of these things are kind of coming together to make it an interestingly difficult problem to solve. But we're kind of implementing different methodologies to try to keep them suspended. They live planktonically, so trying to keep them up in the water column to prevent mechanical damage. But yeah, a lot of interesting problems from what seems to be a highly invasive animal.
Yeah, this is a really good question that just came in. So this is the one I'll throw to you next. But this person was curious about implications of your work for native mussels and native mussel research and work, perhaps at the zoo, perhaps at the DNR, perhaps elsewhere. Do you anticipate or expect or already know of ways that this will benefit native mussel work?
Yeah, the other part of my job, I work over at Minnesota Zoo doing native mussel propagation for restoration in partnership with the Minnesota DNR. And in that, similar strategies of using algae for the very young kind of larval, post-larval stages of those mussels. And it's one of those things that's kind of a how much effort do you put in for what results you get. And with a lot of our native species, once they've transformed off of the fish, actually do pretty well with a mixture of lake water and then some of those prepared algal diets from like reed mariculture, Nanochloropsis, which are also saltwater species. One of the major actual benefits is I do spin down those concentrated marine algae that I use for native algae to get rid of the saltwater content in them, which has shown a little bit of some improvements. But then also a lot of institutions do culture their own freshwater algae for those native mussels, but it's extremely costly with time and then just resources. So if you can grow native mussels just on prepared diets or even just using lake water, that seems to be kind of the best way to go in terms of your cost of input. But that said, I do, if we have extra algae, I do bring it back and feed it to the native mussels when I have a chance. It's a special snack for them.
Yeah, leftovers. Some of us love our leftovers. I kind of a related question here on the flip side. So that was sort of the tie-ins between this work and perhaps the native mussel work that's going on. What about the tie-ins to other AIS research that may be going on here or elsewhere?
We talked about one of the things that would be a huge leverage point for the University of Minnesota in general is if we can complete this life cycle and had a lab where you could be providing um, zebra mussels at various stages of their life to test, biocontrol methods that might be only effective at that, just larval stage or at the adult stage. Right now, there's a kind of a time limitation for when you can do research on zebra mussels because you just collect them in the spring and you've got the summer and then they start to die off. And so if we can keep animals in the lab all year long, we can extend those testing periods of time. as well as if we can produce animals that are at a specific life stage. We have partnerships at the St. Anthony Falls Stream Lab, and then other labs, throughout, um, well, throughout Northern America, that would benefit from being able to receive larvae at a specific stage, that were all from the same parentage and same cohort, would give a lot of controls to a lot of those other experiments. So yeah, there's a lot of benefits if we can get this working really well.
Great, thank you. I've got another question here, and we have time for at least another question or two, so please keep your questions coming here. I'll throw out this one that I'll sort of paraphrase, and I think it's a great question that I think we should all be being asked and being forced to think about. What would you say are the most important sort of brand new insights that have come out of this so far, at least from your perspective, or at least things that maybe surprised you that you weren't expecting to encounter, or that really stumped you at first when you encountered it?
Yeah, one of the things specific to this particular aspect of this project, this algae rearing, anyone who has a pet, you think about what you're feeding to your cat or dog, pretty selective about that food source because that has significant impacts to the health of that animal. So you think about what goes into that food, and in terms of this algae, it's uh, the algae is the food that's going into the zebra mussel, but we're also thinking about the food that the algae are eating, and so that's been interesting to kind of piece apart. There's very specific media, kind of recipes that go into feeding algae, and so trying to figure out the correct one. If there's any sort of limitation from one particular aspect of some sort of a nutrient, in some cases actually is beneficial to the algae, because it overproduces different lipids, especially some of the essential fatty acids, which can be broken up to create other fatty acids. So we found that we're trying to identify what the specific lipid profile of our specific strains of algae are now that I switched the Chlorella vulgara to a different media. So this tap media that we're growing it on, which nobody had really known if you could grow it on that. And so I'm interested to see what those stresses might actually have implications for better nutritional profiles trying to feed them. So that was something that I'm not a nutritionist by trade. Always thinking about nutrition and the care of animals. I grew up most of my career as an aquarist, but yeah, trying to see that firsthand about how there's those implications and how you might utilize certain aspects of stress to improve the project overall. It's been kind of an interesting thing I've stumbled upon.
That's very cool. All right, for the interest of time, I think we'll end this part of the presentation here.
I wanted to start out by thanking the group for choosing this project that is focused on genetic control of grass carp using RNA interference. I am Jim Wamboldt. I am a fish biologist at the Upper Midwest Environmental Sciences Center in La Crosse, Wisconsin, and my co-PI is Chris Merkes, also at UMass, and he's a geneticist. And this project is founded on the combination of my experience working on aquatic invasive species control through the use of baits and attractants, and Chris's background in molecular biology for monitoring and control of aquatic invasive species. Because Minnesota is not a closed system, and with what we've seen historically as invasive carp is spread throughout the Mississippi River Basin, a bit of information outside of the state is necessary to understand our local grass carp situation. And while grass carp are prohibited in Minnesota, many of the neighboring states to the north, you can see that similar as well. But downriver in the map on top, you can see that there's a lot of states that they're not prohibited, and the use of both diploid and triploid grass carp are allowed. In addition to the distribution of grass carp, natural recruitment is known to occur in many locations just downriver of Minnesota. And this is an issue because grass carp will directly affect the local ecology once introduced by reducing or even eliminating macrophyte communities that have disrupted native species and their food webs, and even impact property through the increased erosion when you lose that aquatic vegetation barrier along the shorelines. Secondarily, this overgrazing has been shown to cause a shift in primary producers and consumers and the nutrient cycling favors a more planktonic community in the absence of macrophytes. This causes an increase in turbidity and detrimental algal blooms. And often, we talk about control of invasive carp as this umbrella term that includes the 4 species of carp that we're talking about. And one of the reasons is this mutualistic relationship that grass carp have with the other species that we include in invasive carps. So this shift from macrophyte-driven communities towards an increase in phytoplankton and zooplankton really only makes the environment more suitable for fish like the bighead and the silver carp. And currently, the only methods of control of an established population is either through physical removal with commercial harvesters or other government agencies, or the use of pesticides. And as I said, because Minnesota is not isolated, we need to look at the situation that's going on in the upper Mississippi River as a whole. And if we look at the lower pools, since 2015, we've seen dramatic increases in captures of grass carp in the lower pools of the UMR. And population models have shown that increased invasive carp recruitment downriver does impact the distribution. And it's important to pay attention to what's going on downriver to help make decisions further up here. And I'm sure many of you have seen the news reports the last few years about the relatively large numbers of invasive carp that we've been seeing being caught and being reported in the upper pools in Minnesota. And many of these fish have been grass carp. And although we don't have complete compiled information right now, but it looks like there was a big push upriver during the spring flood in 2023, including a large number of radio tagged fish that came up from pool 15 that have been detected by Fish and Wildlife in the Minnesota DNR that have real-time receivers out in pools up here. This was also the first year that a large number of jumping fish were seen and reported below dams in Winona. And my background has focused on 2 main areas since I've been here with USGS. I've been developing baits that can be used to attract fish to targeted locations for physical removal, as well as incorporating control chemicals into these baits to increase species selectivity. And our group has worked to develop a common carp bait to deliver a lethal dose of annamycin, as well as a microparticle for delivering a control chemical to filter feeders like bighead and silver carp. And the last few years, we have also been testing a non-toxic bait in the Lake Erie Basin to enhance removal efforts that are being undertaken by the states out there. And this work is going to be moving towards Pool 19 in 2024 in the field. So starting in 2019, my team developed a method for quickly and quantitatively evaluating bait formulations to test different ingredients. Ultimately, this led to the grass carp bait that we've been using in the field, as well as our toxicity trials conducted here in the lab. And this method allows us to evaluate the bait as a delivery tool for different chemical controls, or in our case, for an RNAi payload. And we can do this by extracting tracer dye out of the fish tissues and determine the bait and its particular ingredients, or the payload in the sense, its palatability, and quantify ingestion. So our goal is to develop safe control technologies for AIS management using genetic controls incorporated into a species-specific bait. And our objectives are to develop methods, protocols, and SOPs for the construction, mass production, drying, and storage of microalgae that we're using as a RNAi payload. We want to determine the optimal inclusion rate of this payload to ensure desired product standards for offering grass carp the maximum double-stranded RNA dose and the stability of that package once it's put in the water. Lastly, we want to evaluate the efficacy of gene knockdown through oral delivery of this bait and the RNAi microalgae payload.
All right, and now I'm going to pick up the presentation here. Thank you, Jim. Just to give the short version of how RNA interference works. If you want to focus on the graphic on the right side of the screen there, basically, if you remember your grade school biology about the central dogma of biology, you have the DNA blueprints of everything in your body that gets transcribed into messenger RNAs. And then those messenger RNAs get translated into protein. How RNA interference works is we design these small interfering RNAs, which would be depicted by the small molecule at the bottom of the graphic, which are complementary to our target messenger RNA. And then that gets incorporated into what's called the dicer complex, or into the RISC complex by enzyme called dicer. And then the messenger RNA gets cut. And so the gene does not get made into a protein. So essentially, you are stopping the function of your target gene. And we like this technology because you can use it like a chemical pesticide, for example, but it's much more specific because non-target organisms could be exposed to this and have no effect on them because they don't have the same target sequence. And so we've been working on developing some silencing RNAs for candidate target genes in primary cultured cells in the lab. And we're targeting 4 genes that I have listed here: basic transcription factor 3, glutathione peroxidase, keratin cytoskeletal type 1, and glyceraldehyde 3-phosphate dehydrogenase. And our hope is that by essentially turning off these genes in the fish, it will lead to mortality. And so we're demonstrating in the tissue culture knockdown of the genes, and then by testing the different candidate RNAs in tissue culture, then we can incorporate that into our RNAi payload that Jim was just talking about. Now we chose these targets based on genes that were highly expressed and have critical functions. And the picture at the bottom of the screen there, that is what the primary cultured gill cells that we're using look like under a microscope at 100x magnification. Next slide, Jim. So as we mentioned, the RNAi payload is going to be microalgae. And how this works is the microalgae will express a double-stranded RNA that contains the small interfering RNA sequence. And basically, the microalgae will serve as our production facilities, so to speak. They'll make many copies of our silencing RNA, and then the fish will eat this algae, and that's how the RNAi will be delivered. Grass carp don't naturally just eat algae, so this will be incorporated into the bait that Jim just described. And as Dr. Minerich just demonstrated so nicely, we can grow algae in the lab, but not on the scale that you would need to apply in the field for getting rid of invasive species. So we've partnered with a private company called Sundew, who will be working with us on this project to develop the manufacturing processes to scale up the freeze drying and preservation and storage of the algae so that it can be incorporated into the bait, and then we can use that for the testing. Next slide. And then once we have some of the double-stranded RNA expressing microalgae, we will do a series of in vivo oral gavage trials using grass carp. And here's an example of some test tanks that we have in our facility down here in La Crosse. And so we will use oral gavage to deliver the bait so that every fish will know exactly what dose gets delivered. Every fish will get the same dose. And we can have good reproducible results, hopefully. And then we will extract RNA from target tissue from samples at 8 hours and 24 hours after treatment. And these times were selected based on some previous gut passage time data that Jim has collected. And then, you know, kind of some understanding about how long it might take to affect the gene expression and how long that effect might last. So ideally, we're hoping to see effective knockdown and measure that before the effect might go away. And then, so we'll measure that using qPCR. And then after some trials, we will be combining multiple of the candid RNAs to knock down multiple genes in the same fish, and then doing a longer trial to evaluate for mortality. Now, RNAi is successfully used in agriculture to control for pests. But in those cases, they're typically knocking down like 4 or 5 target genes. So we're not necessarily expecting lethality for individual one single targets. But because we're targeting critical genes and because we're selecting effective candidates based on some preliminary cell culture testing, we're hoping that we can see some mortality with 2 or 3, or depending on what our results are from the original or from the first trials, that will inform our follow-up trials. And we'll see how that data guides us for the additional trials. Next slide. So our expected results are to have an optimized method for the microalgae production. And then we'll have optimized bait recipe, demonstrated proof of concept for orally delivering an RNAi for knocking down grass carp genes, and hopefully demonstration of lethality as well. Potential users will be state and federal management agencies, potentially tribal as well. Minnesota DNR is currently reviewing genetic biocontrols, but hasn't yet taken a position on its use. Obviously, this is a new and emerging field, so the ethics and understanding of it has to be carefully thought out, as well as the regulatory side. Future directions, we have to do a lot of ongoing work to evaluate not only just the efficacy, but the safety of this. And also, you may have heard David Fulton's talk in the Web Room 2 last hour on attitudes toward RNAi methods for controlling invasive carp. We're also collaborating with him to get some of the social science understanding about this technology to help guide some of our future work and how it might be applied and such. And then because this is like a genetic based method, but some of the practical application side of things could be widely applicable, but just slight modifications of the genetic sequence, there's potential to apply this to a broad range of different species that are concerned in Minnesota. And we're looking at also using it for other species as well. And then we're also going to be definitely looking at registering this as a biopesticide with the EPA once we have all our ducks in a row and have things ready to go. Next slide. And so thank you everyone for your time, and we're looking forward to working on this project.
All right, thank you, Chris and James, for that presentation. We have a few minutes here for some questions. So I'll remind everybody that you can use the Q&A feature here in Zoom to ask those questions. We have one here that I think is a good one that we can start with, Jim and Chris, and we'll have to be mindful of time because we only have about 4-ish minutes here, but I think that should be okay. Someone was wondering about specificity and the process for hunting down candidate genes, because they acknowledged, the goal is to create something that's really targeted, but obviously fish are related to each other. So there's going to be some conserved genetics there. And so how do you go hunting for candidate genes that are going to be meaningful to impact, but are unlikely to run into any specificity challenges?
That's an excellent question. And to be honest, when I first started working on this project, that was a major concern of mine, because I wasn't sure how critical of a gene I'd be able to go after, because typically more critical genes are more conserved than less critical genes, just because of evolutionary constraints. But as I started doing some of my target selection and looking at sequence alignments between the invasive carp and other related species and some of the native species that might be cohabitating the same waters, I'm finding that even some of the most crucial genes have hypervariable regions that could be very good areas to target. Sometimes it doesn't work out that those are not good regions for designing effective silencing RNAs. So, for example, my initial screen, I had 5 candidate genes, but one of them had too much sequence homology to some of the native species, so I had to drop that from my initial screen. But I'm finding that there's enough variability that I can design some good RNAs, and we're going to do very thorough testing. So we'll have empirical data to show that it's selective.
Very cool. All right. So I think this is another good question, although we'll have to do the brief version, but sort of in a minute and at a general high level, how would you differentiate between this RNA interference and genetic biocontrol and maybe the other ways that people might think about it?
Well, I think that probably the most basic way to differentiate is how long these methods persist in the environment. And so some methods are self-propagating, like a gene drive, for example. Some methods will persist in the environment for a number of years, like some of the things that Dr. Szymanski is working on with reproductive incompatibility. And then this is kind of on the lower end of the spectrum, where you'll put it out in the environment and it will be taken up or eaten, and it'll degrade pretty rapidly and it will not persist. And so it's kind of on the lower risk side from an environmental perspective, is how I look at it.
Cool. Thank you. Thank you to you both. For the interests of running a tight ship and keeping things on time here, I'm going to end us there. I hope everybody will sort of join me in thanking Ben and James and Chris for presenting their work here today.